Hi, everyone. I'm Bill Bosl. I'm primarily affiliated with Boston Children's Hospital and the Computational Health Informatics program. Boston Children's Hospital is a pediatric teaching hospital for Harvard Medical School. I also hold a faculty position with the University of San Francisco. The title of my talk today is A Data-Driven Approach to Measuring Conscious and Unconscious Brain Function. Now, this talk is relatively short, so we don't have a lot of time to go into details. So I'm going to try to just make three important points. And I have a modest proposal at the end. I am not a consciousness researcher at this time, but I'm doing some work that I think might be relevant to consciousness research, and I'm quite interested in looking for collaborators. The primary focus of my research is on developing digital biomarkers for brain disorders, primarily in children, but it's applicable throughout the lifespan. The three main points I want to make are first, that the brain is a dynamical system. And by this, I mean a dynamical system in the mathematical definition of a dynamical system. Because of this, mathematical theorems and concepts that have been developed by the mathematical community apply to the brain. The second main point is brain function can be reverse engineered by phase space reconstruction. I'll talk a little bit about what that means, but essentially it says that time series measurements of a dynamical system can be used to determine functional properties of that system. Since the brain is a dynamical system, those theorems and the phase space reconstruction methods apply. In common terms, what that really means is that EEG measurements contain a whole lot of information about brain function, and far more than we ever thought. The third point is that machine learning methods and feature extraction methods can be used to map the abstract phase space numbers that we derive from this reverse engineering process to clinical measures that we care about, measures of consciousness or descriptions of consciousness, and by so doing, map brain function or neural function to the clinical characteristics that we care about. And then I have a suggestion for a potential data-driven program for consciousness research. So first, the brain is a dynamical system. Dynamical system, many of you may know already in considerable depth. It's a system composed of components or variables that change over time. Moreover, the variables are coupled together in some way. So originally, dynamical systems were studied as sets of differential equations. Neural systems, in fact, are modeled using systems of differential equations. So in a very direct way, neural systems are modeled in the same way that dynamical systems such as planetary motion were modeled. The complex, possibly fractal network structure that comprises the brain gives rise to complex electrical signals. I've showed some illustrations here. The brain, we know, is made up of a neural network that is not randomly connected at all, but has a very specific structure. It's thought by some to be fractal. That's maybe a bit controversial, but it certainly has a very definite structure. That structure gives rise to signals with very particular properties. They are not random. They are at least partially chaotic. And that gives us a lot of information about how to uncover brain function. I've mentioned briefly, or showed a picture briefly in the last slide of a phase space trajectory. Phase space is generally an abstract representation of the state of a system. And by state, I mean the characteristic of each component of that system at any time. If we are looking at a 2- or 3-dimensional system, then we can make a 3-dimensional plot of the trajectory of that system, which may be a particle as in this illustration here, showing how that particle system moves in time through space. Time is not explicitly shown in this phase space portrait. Rather, it's implicit that the continuity of the trajectory shown implies continuity in time. The trajectory shows a sequence of states, so at any point in this three-dimensional representation, the particle has a specific state. And this portrait then describes all the dynamics of the system. It shows how the particle moves in time and what its possible states are. We might consider consciousness and unconsciousness, and I'm making a big jump here to neural systems, that there are phases in the state space of neural function. That requires that we consider the brain, that is the set of neurons that make up the brain, to be a high-dimensional system that can be represented in a very high-dimensional phase space. But we can consider an abstract phase portrait or phase trajectory of that system, even if we can't visualize it. In fact, brain measurements don't have to be visualized to be useful. We know that progress in modern physics was enabled when visualization was abandoned in favor of abstract mathematics. Quantum theory, general relativity, they couldn't have emerged without the abandonment of visualization and adoption of essentially a trust in abstract mathematics. So we're proposing a somewhat analogous approach to describing brain or neural function. And I've shown schematically here this complex system of neurons might be represented by, on the order of 10 to the 11th, roughly 100 billion neurons in the brain. And if we could describe the state of that neuron at every moment, then we would have a representation of the state of the entire neural system. We won't get into details about how to represent the state of a neuron, its spiking rate, its potential. There are a lot of different ways, but without loss of generality, we'll assume that we can describe its state in some way. So, jumping now to phase space reconstruction and embedding theorems. These are results that mathematicians derived over the last 2 or 3 decades, so relatively recent. And the implication, without going into too much detail, is this: that a time series from a single or linear combination of components of a coupled dynamical system, and remember we posited in the beginning the brain is a dynamical system, that a single time series contains sufficient information to reconstruct the phase portrait for the entire system. This is profoundly important for EEG measurements. That says that the EEG sensors measuring time series of cortical electrical fields contain information that can be used to reconstruct the phase portrait of neural dynamics. Not 10 to the 11th neurons, but neurons tend to act in very large groups, so the dimensionality is much smaller. There are a lot of caveats here, of course. These theorems assume a continuous measurement of time series, and of course, today we're measuring digital signals. There's an assumption that there's no noise, it's a perfect measurement, and so on. Despite all this, the implications are that EEG measurements contain a great deal about brain function, and that can be discovered by phase space reconstruction. This is a summary of what I just said, that the complex network of neurons that comprise a brain meet the definition of a dynamical system, and therefore the methods for characterizing dynamical systems mathematically are applicable to time series derived from brain measurements. Well, these methods have developed rather rapidly in the last 15 years or so as personal computers have become available to scientists for experimenting. And methods for deriving abstract representations of a phase space from EEG time series have developed. There are a number of different methods for phase space reconstruction. We can't go through all of those, but they involve computation on multiple scales or multiple frequency bands of quantities such as entropy, Lyapunov exponents, correlation dimensions, Hurst exponents. There's a whole series of nonlinear measurements that say something about the phase space dynamics. There are some newer methods as well. Recurrence quantitative analysis, which is an effort to statistically describe a recurrence plot, which is a two-dimensional projection of the abstract phase space portrait. And even newer, recurrence network analysis, which involves mapping a recurrence plot from a time series to network structures and then computing various network properties. All of these different measurements say something about the dynamics of the system from which they were derived. We have been pursuing an experimental approach or a data-driven approach to extracting what these mean in the context of digital biomarkers for brain disorders, as opposed to trying to figure out a priori or theoretically what these mean in terms of neurophysiology. We're simply looking at data, a set of numbers that represents the abstract phase portrait of the system. Well, we simply measure or get the measurements from patients into clinical groups that have some characteristic of interest to us. And I'll give two examples in just a moment. The two conditions we've been studying are autism and epilepsy. We're moving into anxiety and some other disorders as well. And I hope you'll see in a moment that this could apply to the study of consciousness. The idea is to measure from groups that share some characteristic of interest to us and compare using machine learning and feature extraction methods the phase portrait numbers that we've derived from each of those groups to find what differs between them. I mentioned autism spectrum disorder. You've probably heard about autism in the news. It's well known that it seems to be increasing. We won't go into why that might be happening. But symptoms tend to emerge somewhere between about 2 and 3 years old. It's thought that if we could detect brain developments that indicate emerging autism before the symptoms appear, that there are ways to intervene to maybe reduce or eliminate those symptoms from ever emerging. And here I said some things about the disorder. In our work, applying this data-driven approach where we map features extracted from EEG measurements to groups of kids, either who have developed autism or do not have a diagnosis of autism. They were recruited in a study called the Infant Siblings Project, where we found children with a diagnosis of autism who had an infant sibling recently born and recruited them into this project. Those kids have a higher chance of developing autism, and so they were a good population to study from birth through 3 years when we could see what the outcome was. The summary of our results was that between 3 and 9 months, and especially between 6 and 9 months, we were able to predict with very high accuracy, approaching 100%, which child would develop autism and which would not. Even more, we were able to predict the severity of their autism as defined by the ADOS summary score. ADOS is a commonly used assessment of autism characteristics. It stands for Autism Diagnostic Observation Scale. This plot just shows dashed lines were the actual 24-month ADOS scores for 3 different groups, low-risk controls, kids who actually developed autism, and a third group, high-risk siblings who did not develop autism but had some lesser endophenotype characteristics of autism. And we found that from the EEG alone, we were able to learn through machine learning algorithms how to predict the actual autism score. Jumping to epilepsy for a moment, our conceptual representation of epilepsy is shown here with a two-dimensional phase portrait. People without seizures, as shown in the far left, there's a rather large gulf between the non-seizure state and the seizure state. And on the far right, the epileptic brain, the phase trajectory of the brain in those cases, and between the two states, non-seizure and seizure, is blurred. The trajectory easily moves back and forth between these states. Why that is something we're not getting into yet. We simply want to know, can we measure and reconstruct phase space from EEG measurements that will tell us who has epilepsy and who doesn't? We're really looking for an early marker for epilepsy. Results from just one of our studies here, a certain childhood epilepsy called Beck's, shows that we could detect while the child is awake, showing no symptoms of epilepsy while they're awake, and from EEGs that to neurologists appear to be normal, we could find the region of the brain that was epileptic, and because its nonlinear properties from this phase space reconstruction process were quite different from non-epileptic controls. And even, this is a focal epilepsy, so it occurs in one region of the brain. We could even tell what region of the brain was epileptic. Similar results for absence epilepsy. I don't have time to go into that now, but it's here in the slides. And just finally, I wanted to mention that we're building a software architecture for carrying out this data-driven approach to EEG measurement, mapping to behavioral or cognitive outcomes through our group at Boston Children's, also collaborating with the Laboratory for Computational Physiology at MIT. This schematically tries to illustrate what we're building to carry out the steps that I've just described. Part of what makes this possible is there's a new generation of EEG devices with dry sensors, relatively easy to use, wireless. I just wanted to mention, we're not building devices, but they're coming along commercially, and they open up this possibility for us. And finally, my proposed research program for consciousness, because we're building this shared EEG database that will be open to other researchers to put data in, we're also building the computational tools for computing phase space reconstruction numbers, if you will, and the machine learning tools to map that to clinical outcomes of interest, and that will also be available to the community. What I really want then are standardized labels for outcomes of interest. So consciousness researchers having data from various conscious and unconscious states, whatever the paradigms might be, can access some of these tools or collaborate with me or others in our group to carry out the kind of research we've done for autism and epilepsy for other disorders of consciousness.
Hi, my name is Ken Kishida. I'm an assistant professor in the Department of Physiology and Pharmacology, as well as the Department of Neurosurgery at Wake Forest School of Medicine. Today, I'm going to talk to you about some exciting new technology that we've developed that allows us to perform ultra-fast monitoring of neurotransmitter micro-fluctuations during choice and subjective experience in humans. Just to give you a brief outline of the talk, I'm going to give you an introduction to my lab and the background specifically to the approach of neurotransmitter monitoring. And then I'll go into briefly describe the first sub-second measurements of dopamine and serotonin in humans and some really exciting results that we had there. And then I'll finish with a brief discussion about how I envision linking these kinds of measurements to subjective experience and related behavior through computational approaches. So we'll start with the introduction and background to my approach. My lab is really fundamentally interested in the problem of choice and how our subjective experiences modulate our choices and how choices modulate our experiences. This is particularly important to a medical school because there's a wide number of neurological and psychiatric conditions that have been described historically as disorders of the will or disorders of experience. Specifically to dopamine and serotonin, each of these images here give you an idea of the importance that they play in human health. And so Parkinson's disease caused by loss of dopamine, depression, impulse control, serotonin reuptake inhibitors because of medications that are often prescribed for psychiatric conditions. All of these point to these really important neuromodulator systems and how they affect behavior and the choices that humans make. So we study this problem through studying behavior, but also pairing that with non-invasive as well as invasive measurements of brain activity. So I'm showing you here some tools that we use: functional magnetic resonance imaging, magnetoencephalography, intracortical recordings performed using stereo EEG during epilepsy monitoring. But specifically today, I'll talk about a novel approach which allows us to measure neurotransmitter release with high temporal resolution. These are invasive measurements. Excuse me. These are invasive measurements and require brain surgery. The method we use to do this is called elastic net electrochemistry. Elastic net electrochemistry is a machine learning-based approach to fast scan cyclic voltammetry, which I'll describe in a little more detail in just a moment. But what we've demonstrated is that we can measure dopamine and serotonin simultaneously from the same electrode in conscious behaving humans during brain surgery. We've recently extended this work to be able to simultaneously detect norepinephrine as well. And we're developing methods in which will allow us to monitor these chemicals from electrodes implanted semi-chronically during epilepsy monitoring procedures. The work I'm going to talk to you about today is performed in patients undergoing DBS electrode implantation procedure for the treatment of Parkinson's disease or essential tremor. In this approach, we're able to safely record from the striatum, either the putamen or the caudate that's shown here, while patients are awake and performing our tasks. So fast scan cyclic voltammetry is a voltammetric method that relies on the ability to drive the electrochemical oxidation or reduction of chemical species of interest. This is achieved by placing an electrode, in our case a carbon fiber microelectrode, into solution or into brain tissue and controlling an applied electric potential. If that applied electric potential is greater than the oxidation potential of chemical species of interest, then we can drive the oxidation of those chemicals, but detect the electron transfer that occurs in that process. So each chemical species of interest has its own oxidation potential, which will allow us to separate dopamine from serotonin from other analytes that may be of interest or of nuisance. So the fast scanning part of this comes from the way that we apply the electric potential. We hold the potential at a low value here, minus 0.6 volts, and then rapidly scan over 5 milliseconds to positive 1.4 volts, and then back down to minus 0.6 volts. This fast scanning part samples a lot of different electric potentials, where we can see here the oxidation of dopamine causes an increase in the resulting current that is concentration dependent. Now, exactly where this peak occurs is going to be chemical species dependent, and that allows us to separate the various chemicals of interest that might be present in the brain. These measurements are repeated once every 100 milliseconds, giving us a 10 hertz sampling rate. Here's an example of what the electrode looks like. This has been adapted to match specifications needed to safely perform this in human brain surgery. So we've been able to use this technology to make direct and fast neurochemical measurements in humans while they perform decision-making and learning tasks in the OR. This is done in collaboration with Dr. Stephen Tatter and Adrian Lapson, are two functional neurosurgeons at Wake Forest, in patients undergoing DBS electrode implantation, except for Parkinson's disease or essential tremor. Here's an example of one patient who was having electrodes implanted for the treatment of Parkinson's disease in the subthalamic nucleus. During this procedure, we're able to reach the caudate with our recording electrodes and measure dopamine while they perform a task as shown here. This particular patient was performing a stock market gambling task, which I'll describe in a little more detail in just a moment. And here, I'll show you a real-time trace what the dopamine response does following two sequential wins. So win 1, the dopamine rises. Win number 2, and the dopamine rises again. So this may lead you to believe that dopamine responds to winning during gambling tasks or other kinds of decision-making scenarios. That's not the whole story. So now I'll go into a little bit more detail about that study and describe to you results from the first sub-second measurements of dopamine and serotonin studies in humans, and then finish up describing exactly how I envision linking these kinds of measurements to subjective experience reports through computational models. So one question we can ask is, what information does dopamine release encode? The long-standing hypothesis is that dopamine neurons encode reward prediction errors, where the pause, burst, and change in spiking activity originating from the soma of dopamine neurons represent what are called reward prediction errors, better than, worse than, or as expected. This is achieved by changes in the spike rate, where a pause, burst, or no change in spiking represent deviations in the expected reward. A pause being worse than expected, a burst being better than expected, and no change meaning that the reward is exactly what was expected by the system. So spikes generated in the soma result in the release of dopamine at downstream structures, which include all throughout the cortex, but also specifically in the striatum, where we're able to record from in these patients. In prior work using functional magnetic resonance imaging, this equation was used to track down error signals throughout the brain in the same stock market task that I'm going to describe in just a moment. Here we see that reward prediction errors are encoded in the caudate, consistent with the idea that this signal, the reward prediction error, is encoding downstream release of dopamine in these structures. However, fMRI does not give you the chemical nor temporal resolution to say this definitively. So the stock market task that we use to study what dopamine may encode is depicted here. Patients see a stock market trace, and it's their choice to decide how much they'd like to invest of their portfolio. So the stock market game that we use to probe dopamine release and serotonin release in these first studies is depicted here. Patients see a stock market price chart, and their decision is to decide how much they'd like to invest of their portfolio, anywhere from 0 to 100% of their portfolio in 10% increments. The market advances, and based on their investment, they will earn a gain, right? What they didn't invest represents a missed opportunity. Likewise, if the market fluctuates down, then their bet times the market change in price represents the loss that they incur. However, on low bets, they've avoided a loss here. As the bets increase, what we call the counterfactual error, the difference between what could have been had they invested all in versus what they actually invested, diminishes. I just showed you a result where 2 subsequent gains resulted in positive dopamine fluctuations. Here's an example where there's a gain and a loss resulting in a positive fluctuation in dopamine and then a dip in dopamine. And again, this seems to be consistent with the idea that dopamine responses are tracking simple gains and losses. But if we look at the average of all the trials in these tasks over all of our patients, we see that dopamine responses in green for gains and red for losses don't differentiate gains and losses on average. The long-standing hypothesis is that dopamine release would encode positive and negative reward prediction errors. But again, when we compare that over all trials, we see no differentiation here. This led us to a new hypothesis that what might have been could adjust the valuation errors encoded by dopamine. This has two predictions. One, surprising gains that might have been better should be reduced in value, and surprising losses that might have been worse should be increased in value. This represents the reward prediction error signal that is modulated by a counterfactual prediction error signal. And one way to think about this psychologically is a kind of regret and relief type modulation in these responses. So the hypothesis stated this way is that the dopamine transient ought to be proportional to the reward prediction error, which is the classic hypothesis, but now modulated by the counterfactual error. Predictions of this model go with the magnitude of the bet. So for large investments, we should see the classic hypothesis revealed, where positive reward prediction errors are encoded by positive fluctuations in dopamine, and negative reward prediction errors are encoded by negative deviations in dopamine response. But as the bet size diminished, the counterfactual error takes hold and encode positive reward prediction errors as a dip in dopamine and negative reward prediction errors as a positive fluctuation in dopamine because of what could have been better or what could have been worse had they done something different. Here we have the predictions of the model, and here we have our data. And so when we sort these by high, medium, and low bets, we see that the dopamine responses track exactly what's predicted by the superposition model. We can go back into those exact same trials and ask questions about what is serotonin doing. And what we see in serotonin is that serotonin is fluctuating in exactly the opposite pattern, where positive reward prediction errors at high bets result in a dip in serotonin, negative prediction errors result in a positive fluctuation, and vice versa when you take into the counterfactual information. Now, this is consistent with the prevailing hypothesis that dopamine and serotonin are opponent neuromodulators. So just to summarize this part, we see that dopamine release does not simply track reward prediction errors, but rather it seems to superpose reward prediction errors and counterfactual error signals. Serotonin release encodes an opponent learning signal. And importantly, the context modulates exactly how dopamine and serotonin is released in humans. One thing that was striking is that these results were consistent with the idea that chemical fluctuations in the superposition model track how one should feel about gains and losses. Now, we didn't track this in these experiments, but we plan to in future work. So I've shown you some evidence how we can use quantitative gains in models to link human brain activity, specifically neurochemical fluctuations, to try and understand exactly how these systems are encoding information. And they seem to be related to how people ought to feel. The models that we're using, I didn't go into this, but are derived from notions of optimality, from artificial intelligence research, but also game theory. And in doing so, we can take a computational theory of how the human brain works and try to link that to brain measurements, but also behavior. So here's some preliminary data that we have where a patient is playing a choice task, receiving rewards and losses, where we can calculate reward prediction errors and various other computational terms. In blue, we have the patient's actual subjective rating, trial by trial, of what they experience in the game, how much pleasure they derived from the gains and losses in the task. In red, we take the computational terms that describe exactly how patients behave and fit a model to try to predict how they would feel. Now, the benefit of that is that we get these terms and these parameters that we can then go back and link to the neurochemical measurements that we have and try to tease out exactly what contributions dopamine, serotonin, and norepinephrine have to the generation of subjective experience in humans. And so this work was done in collaboration with a number of folks at Wake Forest School of Medicine, but also Virginia Tech. My lab is growing, and we'd like to add to that. So if there's any potential postdocs in the audience that are interested in this kind of work, please reach out to me. You can contact me here via email, follow me on Twitter. Our work is well funded, and we have a lot of work to do. And so again, just if there's any postdocs or postdoc candidates that are interested in this work, please reach out to me. Thank you for your attention.
Good afternoon. My name is Vincent Zarcone. I'm a retired faculty member at Stanford's Department of Psychiatry. This talk will be divided roughly into 9 sections. The first, I'll define the concept of concepts. The second, I'll discuss the development of concepts and associated words, and then pass briefly to some current neuroscience of concepts, and then the need for a typology of concepts and higher order concepts, followed by Carl Pribram's concept of attention. Then I will speculate slightly and present possibilities for new studies of concepts. Next, turn to current methods of analysis, which do not seem to recognize the great rapidity and immense connectivity of concepts and associated words. This will be followed by a brief discussion of Bohm's analogies, which suggests a quantum mechanics underpinning of a matrix of concepts. And finally, I'll close with a philosophic note on Wilfrid Seller's concept of persons, which brings up the limit to our explanations and predictions. First of all, the definition. Concepts are dynamic, frequently revised processes that are widely distributed and connected in the brain. They are neural events that organize information about the world and its regularities, the self, and others. Some concepts are associated with semantic meanings, but none are fully constituted by them. Semantic meanings may connect concepts, but they cannot say the content of conscious percept concepts or other types of concepts. They can only partially represent them. As Cohen and Stewart say, words act as data compressors, making it easy to think automatically about concepts without perpetually having to remember exactly how the concepts work. Concepts are commonly thought of and labeled as images, representations, or engrams, but these labels should only be used as shorthand for what Wilfred Quine calls place markers. They are intensions of general terms that vaguely describe phenomena for which we have no scientific theory. The simplistic notion that concepts are images, engrams, etc., at specific sites in the brain is the creation of a substance out of a substantive. That is, the reification of intentions of general terms. Concepts and associated words and sentences are part of many functions. They tie functions of many different structures together. They provide information in each parts of cycles of perception, memory, prospection, leading to action. The brain does not develop concepts so that it can talk to itself. Rather, concepts are at times part of the brain, preparing the person to communicate intentions to someone in the past or in the present, or both if the communication is overdetermined. Concepts in brain development. Perhaps the best approach to a theory of consciousness, that of Edelman and Tononi, suggests that concepts and words associated with them develop out of the perceptual categories of primary consciousness. Infants and toddlers seem to have what we call proto-concepts with semantic meanings. Babbling and seemingly pointless behaviors evolve into patterns. The child bangs spoons, makes pointing gestures which are interpreted as commands. The child who wants to cuddle seems to have a concept of what is wanted. Later, when a child discovers the impacts of saying no, it is possible to imagine a sense of autonomy and differentiation of concept of the self from the concepts of other events of the world and of other persons has taken a great step forward. The authors have given an important start in the theory of the early stages of development of concepts and semantic meanings. However, they are very clear they offer no theory of the later stages of what they call semantic bootstrapping. In a footnote that should be memorized by all who contemplate a theory of consciousness, they ask the question, did I see red before I learned the word red? The question suggests how difficult the development of a theory of consciousness, concepts, and semantic meanings will be. That it will be immensely more difficult is suggested by another question. Does a 2 or 3-year-old child know, remember, and think about, i.e. introspect, he hurt my feelings, before she knows the meanings of the words? Two very different questions are suggested by the preceding paragraph. The first is, when and where in the brain are neural events that make the acquisition of concepts and the words associated with them possible? The second is, what are the neural events that just are the concepts and semantic meanings associated with them? We cannot begin to develop a theory of the latter until data to answer the former begins to show patterns with the requisite logical and mathematical multiplicity that can lead to models. Data pertinent to the first question is accumulating rapidly and has been accumulated by new methodologies and an actual neuroscience of language. Imaging studies make it increasingly clear that word meanings are acquired and remembered in systems which are related to actions and activity in specific brain regions. At the level of entire languages, graph theory and computer science have enabled machine translation of language and proto-lifelike interactions with artificial intelligence programs. So far, these studies of systems of words and their relation to brain activity are about words for entities in general terms that can be subjects of rational inference in both common sense reasoning and in science. They see correlations and causal pathways between the acquisition and retrieval of word meanings across individuals and language communities in health and disease, with the findings of studies of cytoarchitecture, gene expression, imaging, and electrophysiology. These future studies of the connection of word meanings and brain activities depend on the effective usefulness of mathematical formalisms, graph theory and systems theory, geometry, differential equations, algebraic topology, etc. The choice of the formalism depends on the models of patterns of data which can suggest the most useful mathematics. This is a leapfrog process in which the move between pure and applied mathematics is derived from data derived from ever more complicated methodologies. It is a process for which there is yet no model. In fact, the brain connections and the methodology used to describe it may be so complex that a way forward is extremely difficult to imagine. Systems of concepts and associated word meanings may exist in many different types of discourse, and there's a large variation across individuals within these discourses. A preliminary typology of concepts and associated word meanings begins with the rejection of the idea that dictionary and early formal language philosophy definitions of concepts are adequate. Concepts are not simply representations in words of entities, properties, and relations. In fact, a recent report indicates that invertebrate brains have a capacity for cross-modality integration and the formation of what can be called very primitive concepts. Obviously, these primitive concepts are without any associated semantic meanings. Fodor and Associates' idea of concepts includes those in an interperceptual circle, the that's of spatial temporal localization during an orienting response, followed by the elaboration of top-down influences that enable the use of words in communication. The development of the ability to create percept concepts, to recognize figure against background, and to see as out of the perceptual categories of primary consciousness and the proto-concepts of the preschooler mentioned above is an area in which much research is needed. The timing of these developments must be described and quantified before correlations from data imaging and electrophysiologic studies can be adequately interpreted. The study of more complicated concepts and language of adulthood can be guided by the fact that concepts and the words associated with them are on a continuum of orders of complexity. All of an individual's concepts and the words associated with them are parts of a large, highly connected, rapidly updated matrix. An individual's matrix is absolutely unique. Therefore, a type identity physicalism is impossible for a neuroscience of language. All we can do now is begin the description of some concepts that have associated words and give a rough ordering of their complexity. The goals of these descriptions are protocols that can advance the neuroscience of language and benefit medical science. Most of an individual's concepts, for instance, those are procedural memories and those are parts of many episodic memories, are relatively simple. Concepts of procedural memories for engineering, construction, agriculture, pricing signals and markets, etc., are of little interest for this presentation, except to point out that the frequent continual use of these types of concepts can lead an individual to believe that there are rule-like procedural concepts that can be guides in affect-laden situations. John Searle, philosopher at Berkeley, gives examples of concepts which have one-word command authority over many sensory motor actions. He says ski to himself when he observes that he is not skiing as well as he knows how. Another example is the concept in words, my bad boyfriend, which can result in a 2-hour continual recitation of reasons to reject a relationship. Another philosopher, Susan Hurley, describes a related phenomenon. She once swam the word no in a phosphorescent sea. A very complex motor act guided by a procedural memory concept, writing words on a page, learned in childhood. Many concepts in procedural memory are used automatically or preconsciously. Ride a bike, drive a car, read a story to a child without paying attention to the meaning of the words. This automatic use can be a problem. Use of the concept myprosody of speech, facial expression, and body posture, which is largely unconscious, can be largely also automatic in social situations that are affect-laden, which results in the speaker often not knowing the effects of his behavior and his prosody on others. Another very high-order complex concept is the automatically highly determined use of attention or effort in the mediation between emotion and motives in highly affect-laden interpersonal situations. This was described by Carl Pribram and others as a process that involves frontal cortex, limbic system, and caudal brain monoamine system interactions. The use of the concept of attention and the words associated with it has its own feeling state. It can be learned by use of different psychotherapies. The feeling and thought, I can make an effort now, require an integration of highly connected neural events in widespread areas of the brain, are signs that a person has gained in self-efficacy and is more resilient. Persons in recovery from an addiction make an effort when they shift from concepts of the self in action when addicted to those of themselves after benefit from psychotherapy. Study of these complex concepts used by the self in action could be the beginning of the description of an immense connectivity of the brain that makes higher consciousness possible. Recent reports show that fMRI signals from both hemispheres' cortical surfaces are highly correlated across subjects with words of a script. This indicates a widely dispersed matrix of concepts. Magnetoencephalography reveals the characteristics of cortical systems for comprehension of word meanings. It is now possible to do cross-cultural studies of cognates of English words in other languages of the words for affects. The concepts and associated words used to mediate between motive and emotion, the development in childhood of concepts of affects, concepts that develop in children learning to cope with frustration and bullying by other children. It is now possible to study the fMRI of Gestalt shifts in the mind's eyes and ears of concepts and associated words learned in psychotherapy from concepts of the self in situations that lead to depression, anxiety, and relapse to addiction. The studies can be done before, during, and at follow-up to help determine treatment effectiveness. Studies during treatment can assess progress and readiness to be discharged from treatment. This kind of study can be done with scripts and videos presented during data acquisition. It differs from the study of functional connectivity before and after treatment in OCD patients because the subjects will be using concepts and words learned in psychotherapy during the data collection. These studies will be the beginning of a finer grained dissection of the phenomena of higher order consciousness. They have potential to benefit patients. Perhaps these studies will lead to a scientific theory of concepts and consciousness similar to those of physics, which postulate imperceptible objects and events and their governing principles for the purpose of explaining correlations among perceptibles. Data sets from these studies will be enormous and complex. Problems of measurement and analysis will approach and may supersede those of physics and the development of general relativity and quantum mechanics. The rapidity of association and revision of concepts and associated words with incoming sensory information suggests the influence of oscillatory fields on spike trains in widespread, highly connected areas of the brain. The rapidity of association of words to sensory inputs, a very low energy but very fast acting force, integrates concepts and associated words as the brain receives sensory inputs and prospects verbal responses. Concepts and associated words form as they come into use. Olaf Sporns, in his Discovering the Human Connectome, calls for improved technology and analytic methods on almost every second or third page. Much more data and improvement in the area of graph theory is necessary. Separate systems of nonlinear, non-simultaneous differential equations may have to be combined to explain the functional connectome at micro, meso, and macro levels. Bohm's discussion of analogs between quantum and conscious phenomena suggests that the matrix of concepts, including the percept concepts of sensation and sensory input, is held together by superpositions of quantum events underlying each event of the matrix. The explicative order is manifest in neurophysiology, but the implicative order guides the creation of the explicative order. Similar explanations have been made by Quine and Bertrand Russell. Habits are specific contents of intervals of the space-time of general relativity. By Wilfrid Sellars, sensations and percept concepts are singularities in the space-time continuum. Penrose and Hemmeroff, and new physics will be required to explain consciousness. And Galen Strawson's turn to a metaphysics that includes fundamental experiential particles that can combine in humans a form of panpsychism. In any event, quantum events can be studied only in the context of their effects on neurophysiology, which can only be measured after the collapse of the wave packet. Presumably, this will require some new theory and technology, which 60 years ago, Sellars said was not on the horizon. It still isn't in 2020. Finally, philosophers of mind, such as Sellars, Hacker, and Sporns, agree that there will be no scientific theory of persons. Individual character, intentions, moral and aesthetic judgments, the use of conceptual thinking in the discourse with others that makes us persons is far too complex. As Wittgenstein said 100 years ago, there is a limit to language and thinking. We will never be able to scientifically read each other's thoughts. The best we may be able to achieve is what Sellars calls a stereoscopic view that joins the knowledge we have of ourselves as persons with the scientific view. Adjoining Sellars believed would be far in the future.
Hi, my name is Mark Valladares. I'm a tennis coach in Kansas City who studies body and brain dynamics in in an effort to both understand body and brain dynamics and in an effort to inform the discipline that I'm in. So my talk is entitled Proprioceptive Feel as a Summation of Oscillatory Resonance. Basically, having connected the flow state of high-performance athletes and musicians, and there's evidence from these studies by Charles Lim on this, with whole brain criticality, we have this idea that the body's perceptive faculty can be primed for a heightened sensitivity to a particular response, and this constitutes a sort of predictive cue for proprioceptive feel. The summation of oscillatory resonance, which is it's the synchrony of these neural oscillations, at the point of the body's response to stimulus, would constitute the vibrational signature of that feel state. It doesn't determine the feel state, it doesn't resolve the hard problem in consciousness, but it gives us a quantifiable basis from which we can maybe describe the constraint of synergies as a result of that predictive anticipatory cue. So the proprioceptive feel is basically induced from global oscillatory resonance patterns, which in turn are transduced from mechanosensory distortions in what I'm calling, and what has been referred to by Turvey and Ingber as the multifractal nested tensegrity grid of the body. The consolidation of vibrational signatures constitutes a harmonization of potential sets of subjective states. So basically, the summation process is assigning a qualitative state to this sensory input that's coming in, and it can be defined as a particular feeling once your perception kicks in, but it can actually happen below your perceptive awareness, and that's what the beauty of studying proprioceptive feel gives us. Coupled in this talk is this idea that the microtubulin polymers, which form the basis of the tensegrity grid, because it's a nested tensegrity structure, they provide, because they're both scalar, they have a base of 13, and tensor, they have this flex and this relax to them. They provide an information processing topology, and so we'll get a little bit into that. The crux of it is that proprioceptive feel, the feel state is something that is occurring at the fine scale, and when you're feeling something, that's kind of where your consciousness is, and there's this toggle between sort of perceptual awareness of the feel state and sort of the intrinsic feel, and proprioception is a good way to sort of study that, because we get posture, we get the subattentional sort of response in the form of haptic feedback to stimulus that can occur before you're actually aware that you've responded. And this is an evidence, and it's easy to study, so it becomes a great domain in which to do science on something like the feel state. What all this puts into perspective is the idea that consciousness is real, we're using it, and the elephant in the room is sort of that consciousness is the statement that I make here, that consciousness attunes criticality. And criticality, you might wonder, why is that consciousness necessary to define? Why attribute agency to criticality? Couldn't criticality just be self-organizing, which is in evidence? And but because consciousness is in the system, because you can't push it off the table, and because biological systems are imbued with criticality across scale, you could also argue that criticality is essentially, attuning criticality is essentially what consciousness is doing. And you see this from the recruitment of proteins in biological systems that are imbued with criticality at a disproportionate ratio. I think that it's 10 to the minus 50, so it's really highly unlikely that we get this array of proteins, many of which are infused with criticality. So consciousness likes criticality, and criticality, the system attuning itself to these critical points where you can get a cascade of effects, becomes really important understanding the process that's at work here. So I'm pointing to one of the fields I'm pointing to is out of the cognitive sciences, a survey of performance studies. There's an anomaly that occurs that the late great Guy Van Orden points to as an indication of this concept of fractal time, and basically what you see is as you look at the body of data, you have these sort of power law distributions, the 1/f fractal noise that appears in the data, which indicates that you're working with a nonlinear dynamical system, and you have to account for the fact that motor control, there's a perceptive anomaly involved with motor control, memory tasks, and it might be linked to the instructional that is given in these sort of cognitive domains when people are conducting these experiments. That's certainly a factor, but what we see is sort of a dissipation of control, and if you don't see that, then you're basically not doing enough studies. So critical states are something that we're gravitating to, and basically what's happening here is that you have this behavior that is established, and as soon as you do, you have this this domain in which mind and behavior are seeking critical points, so that you do have an interplay of the control of these changes. So small inputs, small stimulus can trigger qualitatively different modes of behavior, and this leads to the concept of these constrained synergies, the body constraining the synergies of the mind, behavior constraining mind. So the mind is not necessarily a governing. So he conceptualizes it as a blue-collar relationship of mind to body, and you know, in which, like I said, consciousness is seeking these critical points that enables these nonlinear dynamics to sort of unfold, and that brings us to bifurcation theory. Typically, the modality of motor control is a top-down. It's understood as a top-down process. The neurons are you know, governing the behavior and the dynamics of muscles. In terms of the neuroscience, like I said, this is fairly well understood, but it doesn't account for. There's some paradoxes here in terms of how to, you know, where is the control actually happening, and the signaling process is so slow with neural conduction that. And these response times are so fast that we basically have to account for a process that is sort of more together than it appears at the time that the inputs are occurring. The fractal physiology, the whole body is a fractal system. You have this inversely proportional relationship to the structure and the size and the number of constituents of a particular system, and that in itself imposes a kind of constraint on the system. And so fractal physiology, criticality, which we just kind of covered, the system seeking these critical points, and then tensegrity, which we're going to get into here in a sec, will really begin to sort of explain how this whole dynamic comes together. And then the statement control is about understanding constraints on different time scales, and this is really kind of the essential piece of the information processing that has been fleshed out by Anirban, and the potential for that to occur at the tubulin level at the fine scale indicates that we're kind of on the right track. So Guy Van Orden and Chris Kello basically institute this idea that behavioral variation is a fractal. Cognition is long-range correlated across time scales. There's a dissipation event basically, that occurs with it. Coordination of brain and body has its basis in metastability, so the system doesn't just achieve a level of control and maintains it, it's always dissipating. And this is kind of what you see, and there's this sort of dynamic instability. The stability provided by behaviors, the instability imposed by the injection of mind. Tensegrity is originally derived from Bucky Fuller. It's like a geodesic dome that you can compress and it pops back out. It has this conformational stability. It's structural integrity from a system of compression and tension, and that's similar to what's happening in the body with the muscles and the bones and the fascia wrapped around it. It's not just sitting there hanging on. The bones aren't just sitting in there, in vague space. So there's this constant tension in the system, a pre-stress, if you will. And Donald Ingber at Harvard really kind of attacks this concept, demonstrating how mechanical forces influence cell destiny, and you get this mechanotransduction at the cellular scale that is virtually instantaneous, basically an acoustic wave, that can determine how genes express themselves based on the tension in the extracellular matrix. And so that gives us a multifractal nested tensegrity structure. So in other words, just as the bones and the muscles and the fascia are all providing this tensegrity structure, if you unpack that, you go to this finer scale in which you have the tubulin as the bones, in some sense, actin, myosin providing compression, the membrane, and so you get this conceptualization of the brain and the body as an excitable media with these sort of interwoven synergies, and so the tensegrity yields this picture of a collection of these synergistic domains, and as you go from hand to limb to body to trunk, and the constraints are coming from the thicker processes, that principle is still in place. And it gives us a conformational metastability, and so the whole system is essentially in touch with itself from the cellular level to the macroorganism. Any distortions in tension anywhere are detected everywhere, and so it has a sense of itself. The proprioceptive field can be defined within that perceptual system as the body being kind of aware of itself and the adjacent environment in response to it, and Turvey develops this idea beautifully. So what we're getting to tie together here is the idea that the microtubulin polymers are propagating mechanical forces, so they have an inherent sense of the pre-stress tension, but as they're also capable of being conduits of oscillations, and this is what they're doing. And these oscillations end up being, in neural systems, end up being associated with processing and awareness. The fact that the tubulin are scalar and tensor are really important. The scalar, they have a base of 13, so basically what they're doing is it's an information processing topology. There are these harmonic permutations that are generated from noise as it comes in, and information gets integrated. This is the work of Anirban Bandyopadhyay, and here you can see him sticking a direct measurement into a probe into a microtubulin bundle and basically reading the activity. And one of the things that happens out of these, when he starts to probe these tubulins, is that because of the resonance patterns, the small amount of energy that is coursing through there, through these resonant frequencies, can yield a greater amount of energy. And so that's kind of an unusual an anomaly that tripped him a little bit, but it was verified elsewhere by this guy named Muller, and so this is really the current of thoughts and meaningfulness having this sort of effect that can scale up. And here we see the breakdown of it into these three bands, and he calls this the triplet of triplet configurations. And here what he's done is he's gone inside a neuron, inside the microtubulin, and he sees these three bands at three different frequencies. And so he calls it the triplet of triplet configuration, and then each of these bands has these 8 sort of peaks. And so the 9 into 8 produces what he calls 72 clocks. and we were talking about how control is about imposing constraints on a fractal time scale. And here we see the evidence surfacing for exactly that. So it's as if as these patterns are coursing through the body, it's understanding the role of the mind and behavior in the context of fractal time. This is a centriole, which also, I throw this in here to sort of reflect how every cell in the body has this, and this is where the tubulins converge and are made, basically. And it's also a triplet. It's a triplet configuration, and so it's theoretical, but it's possible that every cell would have access to those resonance patterns. Intrinsic feel and qualia have to do with this is a study from Ross that I'm pointing to indicate how the beta band, which is very in strong evidence in association with motor behaviors from Parkinsonian studies, but proprioception in this study or haptic feedback is correlated with beta synchrony because of the time frame. The onset to steady state is just 10 milliseconds, which is the amount of time that you can respond to stimulus as it comes in. And you see this in tennis where people are, will respond to a ball below their awareness of it. It happens quicker than you can sort of perceive. You're responding to it, and then there's a perceptual awareness that's tagged with gamma synchrony at a half second or 400 milliseconds. The intrinsic feel is essentially the pre-stress state of the biotensegrity grid. So the grid has this pre-stress that defines it. So deviations in tension will alter the field. If those deviations in tension, if those distortions in tension are significant, then you get a qualitative change in the state. So all of the input coming in are, in some sense, potential subjective states. And then as it achieves synchrony, you have this, you basically have this perceptive anomaly, a singularity event in which you're perceiving it a certain way. I want to end with this brief discussion of the Trapinski gasket and how it influences, and this will help us understand how energy dynamics at a fractal scale are sort of at work. Now, the Trapinski gasket, we don't know if it's in the body or not, but this is just to illustrate fractal scaling, and basically this is a triangle in which you systematically remove these equilateral triangles from it, and then you get an infinite amount of perimeter as the area diminishes. And so this is a nice way to conceptualize kind of the anomaly of what we see when teaching tennis. So if somebody's learning a skill, basically, there's a tremendous amount of energy that goes into creating this picture of the stroke. You probably break it down into three parts: the setup, the hit, the follow through. And then as the person practices that, they begin to anchor to one piece of the swing that will pull all these chunks together. And then as they continue to do those iterations, the energy then becomes more of a feel state. So then as it goes to the feel state, what we expect is to see less use of area. So it will disappear out of the bigger triangles, and the information about the swing will be dispersed as a feeling. And that's eventually what the forehand becomes and the serve becomes. So that's kind of how I'm tying this to tennis. Thank you very much. That's the presentation.
What's up, everyone? My name is Isaac Christian. I am a current first-year graduate student at Princeton University, and the research that I'll be presenting today is from San Diego State in Dr. Jillian Lee Wiggins' lab. So to begin, I'd like to start off by posing the question, what constitutes a cognitive state? So right now, when I have your full, undistracted engagement in this presentation, hopefully, of course, is that its own state? The diverse range of cognitive faculties you use, such as language comprehension, visual processing, all coalesce to form this experience right now. And is that state, say, different than a meditative state, where you have the same cognitive functions available to you, but perhaps apply them in a unique way to create a different sort of conscious experience? Or let's say in the unlikely circumstance that during this presentation, your mind wanders off to consider how the weekend's plans are shaping up quite nicely, how you are excited to participate in a local, socially distanced gathering, and you have been given the task to prepare a dance because it's a dance competition. But for the music, you can't decide between your two favorite artists. Is this mind-wandering experience consistent across all your various mind wanderings? And is mind wandering itself separated distinctly from our previous examples of a meditative state or a state when you are engaged in the task at hand? So mind-wandering meditation task states, as well as other conscious states listed here, are a few contexts of growing interest in the literature. And this leads us to further question, what are the other dimensions of experience? How do brain patterns vary across these cognitive states and give rise to the complex and unique ways in which we experience the world? Additionally, what about psychiatric disorders? Is each cognitive state, ranging from watching a movie to mind wandering, processed differently in those with psychiatric disorders compared to typically developing individuals, or TD? And can we identify neural patterns, say, between anxiety and depression that are unique to the diagnosis, but also unique to the state? So how do we measure what's going on underneath the hood? The neural activations that give rise to our experiences. Well, fMRI is an especially powerful tool used to measure bold activity, or varying levels of oxygenated blood flow to the 86 billion neurons in your cranium. From this signal, we can calculate functional connectivity to indirectly measure how information is exchanged between brain regions. So there are multiple methods for calculating functional connectivity, but one that is commonly used is simple correlations, or R values, which measure the similarity between courses of, say, brain regions A and B. If signals from both regions are fluctuating over time in the same way, regions are thought to be connected and exchanging information with one another. Conversely, regions with vastly different activation patterns are anti-correlated or not connected. Using this model, we can identify interactions between brain regions or networks that differ in response to certain stimuli, that vary between groups of individuals, or that are specific to a cognitive state. Okay, so now let's dive deeper into our first question regarding what constitutes a cognitive state. The literature points to two major brain networks whose functional connections are responsible for differences when we are engaged in a task, like listening to this presentation, and resting, or allowing our minds to flit and float and wander at will. The first major network, the lateral frontal parietal network, depicted on the left side, consists of more lateralized areas in the prefrontal and parietal cortices, and is often referred to as the task positive network. The second is the default mode network, the DMN, comprised of more medial areas of the brain. So to further investigate the nature of these state-dependent networks, Keenan and colleagues asked participants to participate in 15 different tasks evoking various cognitive functions. They found that in all tasks, nodes of the LFPN were highly functionally connected, as seen with the average activation across tasks depicted in the center of the circle on the left. And additionally, they found specific variations in how the LFPN was connected to other areas of the brain, depending on the nature of the task, seen in the activation patterns on the outside of the left circle. During rest, however, the medial regions of the prefrontal and posterior cingulate cortices are activated, and critically, the LFPN is not. This relationship is exemplified in Fox et al. Study on the right, where regions of the DMN are highly correlated with one another during rest, displayed in the warmer colors, while regions of the LFPN are anti-correlated with the DMN, depicted in blue. So from this information, it is reasonable to infer that these two major brain networks and the functional connectivity patterns within and between them play major roles in orchestrating the task and rest states. In autism spectrum disorder, or ASD, functional connectivity analysis has been applied in a wide variety of task contexts. Findings of hyperconnectivity or greater connectivity in ASD relative to TD, displayed on the right, and hypoconnectivity or less connectivity relative to TD, seen on the left, have been observed between a variety of brain regions in a number of these cognitive tasks. However, there's been no consensus in finding consistent brain patterns indicative of the condition. Perhaps one of the most studied contexts in ASD are those that elicit socio-emotional responses, as social dysregulation is a key behavioral component in autism. A critical pathway that contributes to socio-emotional processing exists between the amygdala and prefrontal cortex, or PFC. The amygdala is a subcortical hub with unique functional capabilities, traditionally well known for its role in the flight or fight response, but also consistently activates in response to emotional phases and in other social settings. The PFC is thought to regulate the amygdala in this relationship to potentially dampen adverse or over-responsive signals. A common task used to test functional connectivity alterations in ASD between amygdala and PFC requires participants to view emotive faces and identify some characteristic, like the gender, of each stimulus presented. So let's call this viewing of these faces a context or a cognitive state, as subjects are required to engage in a goal-directed task involving emotional stimuli. Two past studies utilizing similar face viewing paradigms found results of hypo and hyperconnectivity between the amygdala and PFC depending on the type of face stimulus presented. Thus, it seems that within an engaged socio-emotional task state, there are connectivity alterations in ASD between the amygdala and PFC, although results for hypo or hyperconnectivity are inconclusive. Resting state has also been well documented in ASD literature. Compared to socio-emotional tasks, alterations between the amygdala and PFC are more consistent in the resting state context, with 3 studies reporting hypoconnectivity in autism relative to TD. So taken together, there is a great deal of literature in ASD demonstrating altered connectivity in a variety of tasks, including socio-emotional ones, and during the rest context. However, experiments are typically designed to test one context, meaning that ASD and TD are given one task in the scanner. By only testing one state, we cannot delineate whether connectivity patterns are due to the varying cognitive demands of the task or a result of the condition of ASD. We therefore created 3 objectives for this study. The first was to discover what brain processes consistently differ between a faces task and resting state regardless of diagnosis. The second was to investigate the brain processes unique to ASD regardless of context. And finally, we wanted to explore connectivity patterns that differ depending on both the context, task or rest, and the diagnosis, ASD or TD. So our hypothesis was we expected alterations to persist between the amygdala and PFC regions in both task and rest-based contexts. We also expected large-scale context-based networks to exhibit unique patterns with the amygdala in both ASD and TD. Our sample consisted of 47 youths with ASD and 72 typically developing individuals. Subjects were excluded if functional scans were unavailable or there was excessive head motion in either scan. 2 separate states were then evoked during fMRI data acquisition. The first was a 6-minute task where participants were presented with happy, sad, neutral, and angry faces and asked to identify the gender of each face presented. The second was a 10-minute resting state scan where subjects were instructed to let their minds wander and not to think of anything in particular. These then were our 2 cognitive states that we tested against each other. Both task and rest data were processed in the same manner. First, raw data from both fMRI runs underwent typical pre-processing procedures. Next, we calculated right and left amygdala connectivity with the rest of the brain, where both time courses from voxels within amygdala masks were extracted and averaged from the pre-processed images, and then correlated with one another with every other voxel in the brain. Pearson R values were then derived from these images and were converted to Z values using Fisher's R to Z transformation, and then inserted into a group analysis where context was within subject's variable and diagnosis a between subjects variable. So our results, yay! In summary, we found connectivity patterns between the amygdala and predominantly frontal and posterior regions of the brain that varied by context regardless of diagnosis, connectivity patterns unique to the diagnosis ASD regardless of context, and patterns that were different between ASD and TD both in the task and rest-based contexts. So in our highest level interaction, context by diagnosis, two significant clusters emerged between the left amygdala and medial regions of the prefrontal cortex. The connectivity patterns were similar for each cluster, where during task, connectivity was higher compared to TD or hyperconnected, while connectivity during rest was less connected in ASD versus TD or hypoconnected. We also found connectivity that persisted regardless of context between the right amygdala and the left insula/superior temporal gyrus region. Here, greater connectivity was observed in ASD compared to TD. And lastly, we found connectivity patterns that were greater during rest compared to task and greater during task compared to rest between the left-right amygdala and a variety of brain regions, including parietal, frontal, temporal, and subcortical areas, regardless of diagnosis. To ensure the validity of our results, we controlled for potential confounds such as anxiety, depression in post-hoc analysis, finding that all clusters still remained significant. So the goal of our study was to investigate how amygdala connectivity varies by cognitive state and diagnosis, and our results indicate that we do indeed find differentiated patterns in each of our three contrasts of interest. This study provides a novel methodology for examining connectivity alterations in psychiatric disorders and further contributes to our understanding of how information processing at the neural level creates variations in the way we experience certain contexts, such as mind wandering and while in an engaged task state. So first, we found that during task, the left amygdala exhibited a greater degree of connectivity with two regions of the medial PFC in ASD compared to TD. This aligns with Monk and colleagues' findings of hyperconnectivity in response to happy versus neutral faces, yet conflicts with Swartz and colleagues' findings of hypoconnectivity in response to sad faces. Our socio-emotional task encompasses a similar design to these two mentioned studies, but differs in the way that we calculated connectivity over the entire task and rest run, and did not directly target how the brain responds to each emotional phase presented. Thus, our results of hyperconnectivity may represent a more common general trend of amygdala hyperactivity over an extended period of socio-emotional processing. Our results for decreased connectivity in ASD relative to TD during rest are in line with all three past studies in ASD during resting state, which report hypoconnectivity between the amygdala and medial PFC. One possible explanation for these findings is that during rest, there is a common baseline of emotional integration, of which the amygdala contributes to. Hypoconnectivity may represent a reduction in ASD's ability to integrate this information in the absence of stimuli. Overall, results here signify ASD processes information uniquely relative to TD, depending on the context. For connectivity that persisted across contexts in ASD, we found a general pattern of hyperconnectivity between the right amygdala and insula. This is in line with past research describing the insula's central role in emotional dysregulation in autism and a variety of other psychiatric disorders involving emotional dysregulation. This further suggests that there may be some brain processes in ASD that are pervasive across numerous cognitive states. Our third contrast found connectivity patterns that were unique to a task or rest state, regardless of diagnosis. Notably, we found the left amygdala to be more connected during task compared to resting state, with more lateralized frontal and posterior regions, including the left inferior parietal lobule and dorsal lateral PFC, which are nodes of the task positive LFPN, as seen here. The same trend of greater connectivity was exhibited for medial posterior regions of the DMN, including the precuneus and posterior cingulate cortex with the right amygdala. Only here, connectivity was greater during rest relative to task. So in both the LFPN and the DMN, we find the amygdala to exhibit greater connectivity depending on the cognitive state, suggesting that the amygdala may be contributing to the function of these large-scale networks. This provides evidence for a unique pattern of socio-emotional processing in each context, where differences in connectivity between areas with more modular functions like responding to faces and more global functions such as adapting to task needs integrate to create differing cognitive states. So varying levels of connectivity with the amygdala and the LFPN may explain why some tasks or contexts evoke deeply emotional experiences while some do not. Additionally, given that these patterns persist regardless of diagnosis, there are some neural processes that exist despite the neurological impact of psychiatric disorders. Furthermore, it seems that there are two distinct pathways between the amygdala and prefrontal parietal cortices, one more medial and one more lateral, that may have different functional capabilities. In an effort to better understand the relationship between the amygdala and PFC, Gangashi and colleagues used neural tracers in the brains of monkeys, finding that more lateral regions of the PFC had more abundant inputs from the amygdala, while medial aspects of the PFC projected or outputted more to the amygdala. It's possible then that lateral regions of, or which receive more inputs may represent the task-positive network recruiting the help of the amygdala during the task. In this way, greater levels of connectivity between nodes of the task-positive LFPN network and the amygdala may indicate greater degrees of socio-emotional processing relevant to the task, such that some participants orient more to the emotions of faces than others. In contrast to lateral connectivity, which may represent this recruiting function, the medial aspects of the PFC may play more of a regulatory role, inhibiting exaggerated and undesirable amygdala responses. This may provide an explanation for social dysregulation in autism or other psychiatric disorders, where alterations in this medial pathway may result in aberrant responses to emotional faces. However, while it is clear that there is a difference in amygdala PFC connectivity between ASD and TD, it is unclear whether increasing levels of connectivity are maladaptive, contributing to behavioral symptom severity, or adaptive, ameliorating such abnormality as an ASD. Or, to put it more simply, are increasing levels of connectivity in this pathway good or bad? The maladaptive hypothesis suggests that increasing levels of connectivity in ASD may reflect rising amygdala activation due to the PFC's inability to suppress or regulate this activation. The other interpretation posits that greater levels of connectivity between the amygdala and PFC may actually be adaptive. And this may actually apply to typically developing individuals as well. So results from a mindfulness-based stress reduction study in adults without any history of psychiatric disorders found greater connectivity between the amygdala and medial PFC when presented with negative affective faces compared to adults who participated in a control program. Mindfulness meditation practice is often associated with a greater awareness and acceptance of experience through a practice of objective labeling, a mechanism which may train emotional regulation. In this way, greater connectivity between the PFC and the amygdala may represent an adaptive effect or a greater contribution, a more willful practice of the PFC in inhibiting otherwise detrimental signals from the amygdala. Studies using other analysis techniques such as effective connectivity should be designed to further test the causal relationship between these regions. So, to review, we found the amygdala, a critical node in socio-emotional processing, is connected differently to various brain regions depending on context, diagnosis, and the interaction of both context and diagnosis. We also found connectivity levels of we found differing levels of connectivity with all three of these conditions that demonstrate the complex yet perhaps consistent ways in which the brain processes information by state and disorder. Taken together, there are distinct neural mechanisms and relationships between those mechanisms that may contribute to the behavior exhibited in psychiatric disorders, as well as the varying ways we experience the world. While the relationship between these connectivity patterns and experiences is not precise nor direct, studies that compare multiple social tasks, such as theory of mind, explicit emotional labeling, should be studied to further inform our interpretations to the question of what is a cognitive state? Where do the similarities lie? Turning to neuroscience is a promising route for understanding why we may intuitively feel that some states, like meditation versus watching a movie, are separate experiences. Perhaps the greater we feel these states to be intrinsically different, the more pronounced the differences in neural patterns would be. Furthermore, an ongoing debate seeks to explain if consciousness is a result of distinct modular subsystems, or a common global workspace? Might these context-dependent networks comprised of multiple co-activated and perhaps conscious regions, along with their anti-correlative relationship with other collective wholes, provide different separate workspaces for experience? Or are there consistent brain regions that are integral in fusing one total workspace? Further studies mapping the relationship between neural patterns and experiences are necessary for better understanding these distinctions. So I'd like to thank Autism Speaks and Michigan Institute for Clinical Health and Research for making this work possible. And I'd like to thank you all for using your LFPN networks and staying with me this entire time. And now, feel free and go forth to mind wander. Thank you.
Hi, my name is Stephen Gubka. I am a PhD student in the philosophy department at the University of Texas at Austin. My presentation today is about the following question: what kind of mental state is an emotion? I'm going to look at two different views to try to give an answer to this question. First view, the Jamesian views, answer that emotions are bodily feelings. I'm going to raise a problem for this view, and then I'm going to go on to argue that another kind of view that treats emotions as evaluative perceptions offers a solution to that problem. In response to this question, what kind of mental state is an emotion, William James famously argued that emotions just are bodily feelings. So take, for example, an episode of fear. Suppose I see a large dog and I become afraid of it. Now, according to James, the emotion itself is going to be preceded by a number of physiological changes. For example, my heart rate is going to increase, my breathing rate is going to increase. James will say that it's the perception or the feeling of these bodily changes that is the emotion itself. Now, naturally, this is a very phenomenologically satisfying view of what an emotion is. It captures exactly what it's like to be in the grips of an emotion, at least what it's like bodily speaking, since we're just talking about bodily feelings here, bodily sensations. This is all well and good, but the Jamesian view suffers from a well-known problem, namely that it cannot explain the intentionality of the emotions. We normally think that emotions are about things in the world. For example, my fear was about the dog. But how is it that bodily feelings can be about external objects? If the feeling of my heart rate increasing is about anything, it's just about my body. How can that feeling alone put me in some kind of representational relationship with the dog, which my emotion is supposedly about? You might think, sure, this theory gets the phenomenology right, but it can't get the intentionality right. It can't show us how emotions are directed at objects in the world, and that's a crucial part of emotion. In response to this problem, a number of philosophers have essentially backed away from the Jamesian theory of emotions. Instead, they proposed that other kinds of intentional cognitive states, like evaluative judgments or thoughts, are just what's necessary or maybe sufficient for having an emotion. So for example, in the case of my fear of the dog, maybe I would need to think something like, that dog is scary, in order to have an emotion about that dog. I need some evaluative judgment or thought, something that's already about the dog, as part of the emotion in order to explain the intentionality of that emotion. I'm not going to talk about these views here. Instead, I'm going to look at a couple of recent, what I'm going to call neo-Jamesian theories that purport to understand bodily feelings in a way that can capture the intentionality of emotions. They can explain how bodily feelings can be related to objects in the world, and so that emotions can be intentional after all, on a Jamesian style view. So here I'm going to talk about three neo-Jamesian theories of emotions. I'm just going to briefly overview them here. I'm going to talk about that in more detail later. And these views all claim to solve the problem of intentionality, make it so that the accounts of emotions they give make emotions directed at something other than bodily sensations. So for example, Dioner and Tironi have argued that emotions are felt action readiness, the bodily sensations that are felt precursors to characteristic actions. So in that episode of fear that I mentioned before, perhaps the felt action readiness for this emotion would be a feeling of being ready to run away. And that would give some more representation to what would otherwise just be a bodily feeling, that it represents an action, being ready for a kind of action in that situation. Griffiths and Scarantino, on the other hand, argue that emotions are also perceptions of bodily changes, but these bodily changes represent affordances or possibilities for action. So the case of fear, perhaps what's represented as possible is just running away. And again, this gives more representational content to the emotion. Finally, Shargel and Prins argue that emotions are perceptions of bodily changes that represent that an afforded action is imperative. So borrowing from Griffiths and Scarantino, they're also talking about affordances or these possibilities for action. But on their view, the bodily changes or the feeling thereof, the possibility that's represented is represented as imperative, as compelled in a certain way. So one doesn't just see that running away from the dog is possible, but one feels that one ought to do it. And again, I'll explain these in a little bit more detail later on, but I hope you can see from this sketch version of each of these how they add some representational content to the emotion and thereby try to solve the problem of intentionality that the Jamesian view had. So here's the plan for the rest of the talk. First, I'm going to argue that emotions are a distinct source of motivation for action and judgment. And this is because I think emotions are reasons indicating, and I'll say a little bit about what that means. Then I'll demonstrate that these neo-Jamesian theories of emotion that I just overviewed can't explain this phenomenon that I just called out, namely that emotions are reasons indicating, make us aware of what reasons we have. And finally, I'll suggest very briefly that a perceptual theory of emotion can explain how emotions are reason indicating. So then we get a nice motivation for discarding the neo-Jamesian theories and thinking maybe we ought to go with theory of emotions that is sui generis and not just reducing them to bodily states or other kinds of states. My worry for the neo-Jamesian views starts with the following question. Why do emotions motivate the judgments and actions that they do, and in such a way that rationalizes those judgments and actions? So take my fear of the dog again. Undergoing such a fear that seems to motivate and rationalize me running away from that dog. At least if we're in good conditions and my fear is accurate because the dog is actually threatening. But why does fear motivate that and not some other kind of response? What is it about fear that does as such? Here's an intuitive answer. Emotions make us aware of prima facie reasons for actions and judgments, and that's why they motivate and rationalize some actions and judgments, but not others. In essence, then, my claim is that emotions are reasons indicating. An emotion, or undergoing an emotion, is sufficient to make one aware of one's prima facie reasons for action and judgment. And that is how I can explain how emotions motivate and rationalize what we believe and what we do. So in the case of my fear of the dog, I'm aware of a prima facie reason to avoid the dog, namely that the dog is dangerous. And without such awareness, it would be hard to explain why I act the way that I do, why that action is motivated by my fear. Someone might worry here that emotions themselves really aren't what's reason indicating. Instead, There might be some other reasons indicating mental state, such as judgment or perception, that's explaining why emotions seem to come along with reasons. And this kind of objection says it's the accompanying state that is giving rise to one's awareness of one's reasons and not the emotions themselves. I'm going to just briefly respond to this objection with 2 points, and that will hopefully also clarify what I mean by reasons indicating. Suppose that my fear of the dog is a recalcitrant emotion, so it's inconsistent with what I believe. And perhaps this is because I have a phobia of dogs. Nonetheless, in this situation, I am afraid of the dog, but I don't believe that the dog is dangerous. I know, either through testimony or my knowledge of dogs in general, that this dog isn't going to harm me, isn't a threat to me, and so on. So my emotion would seem to be inconsistent in some sense with what I believe in the situation. All of this just goes to demonstrate that emotions, even recalcitrant emotions, are still reasons indicating, making one aware of one's reasons, even if those reasons are ultimately irrational or unjustified. And that's just because when I'm undergoing this fear of the dog, it's still going to seem to me as if I have a reason to run away, even though I know I really don't. And that indicates that the emotions themselves are sufficient to make one aware of one's prima facie reasons for action and judgment. And it's not the case that one needs to have some belief also to explain why one has a reason to run away in this case, or to perform any other sort of action in the case of the emotions. Another reason to think that emotions are reason indicating is because of what I call emotional realizations. Now, emotions are often credited with helping us come to realize or believe things that we were already arguably in a position to believe or to know. So for example, when someone says, I didn't realize that what I was doing was wrong, but then I felt guilty. That seems to indicate that the guilt played a crucial role in helping someone realize something that they were probably already in a position to know, right? If they had just thought about what they had done, they might have been able to figure out what I was doing was wrong, but perhaps they didn't really realize what they were doing was wrong until they felt guilty. And this suggests that emotions have to be reasons indicating in order to help us come to realize such truths. And if they weren't, then it's unclear how emotions could play this kind of role where they help us come to grips with certain kinds of normative truths, independently of our reasoning about them. So just cataloging what I take myself to have done so far, I've argued that emotions are reasons indicating, and cases of emotion without a belief and emotional realization demonstrate that emotions are sufficient to make one aware of one's prima facie reasons for judgment and action. So now I'm going to go on to argue that the Neo-Jamesian theories that I talked about earlier can't explain this fact, can't explain how emotions could make us aware of our prima facie reasons for judgment and action. So recall Dionne and Tironi's view. They think that emotions are felt action readiness. So again, taking the case of fear, the feeling of being ready to run away from the dog. And this is how the dog gets into the representational content of the emotion on their view. The action that one feels ready for includes or makes reference to the dog. So all well and good for some theory of intentionality of the emotions, but runs into a problem with whether or not the emotions are reasons indicating in the way that I've argued. Feeling that one's body is ready for action does not indicate a prima facie reason for that action. So after all, that one is ready to undertake some action is not an impaired consideration in favor of that action. Right? So I've got my hand on my mug right now, although you can't see it, and in some sense I'm ready to dash it against the floor. And I take it that that fact is not a consideration in favor of me dashing it against the floor. being ready to do something is not necessarily or even typically a reason to do that thing. So it's unclear how, on Diona and Tironi's view, emotions can properly motivate and rationalize actions just by appealing to the felt action readiness. So recall again the other view we discussed about affordances by Griffiths and Scarantino. They argued that emotions are perceptions of bodily changes that themselves represent affordances. Suppose in the case of fear that it represents the possibility of running away, in this case running away from the dog. Again, we can see how the emotion gets an object in the world, namely the dog, relative to what the action is about, what the action is a response to. So all well and good again for intentionality, but similar problem arises. That one's body represents that an action is afforded doesn't seem to be a prima facie reason for undergoing that action. So something's being possible in general is not usually an impaired consideration, right? So it's possible for right now for me to smash my mug against the floor, but of course that alone, it's the possibility of such an action, is not an impaired consideration in favor of me doing that action. So again, it's unclear how on Griffiths and Scarantino's view, this Neo-Jamesian view in general, if what we're talking about is representing affordances as a part of emotion, how it is that mere representation of affordances can properly motivate and rationalize action. Finally, we have Shargel and Prinz's view that emotions are bodily feelings that represent that an afforded action is imperative. Like the previous view, this idea that an action is afforded relative to some object gives us a clear sense of intentionality with the emotions. And furthermore, we get this idea that the afforded action is represented as imperative. And this is as close as I think we get to a motivational picture on the Neo-Jamesian views. However, I still think it falls short. We can begin by wondering, well, how exactly on this view does one's body represent that an afforded action is imperative? They say bodily perturbations that motivate because they push and pull us. So maybe this is something like feeling discomfort or pain in a similar way that those are bodily sensations that motivate. Perhaps also emotions involve discomfort or pain of a kind that push and pull us. And these are clear candidates, bodily perturbations, for being prima facie reasons for some actions. Take scratching an itch, or stretching, or avoiding something that's too hot. But the question I have here is, how can these bodily perturbations really be reasons about the objects of one's emotions? To take an example here, remember again my fear of the dog motivated me to run away from the dog. On Shargel and Prinz's view, when I feel a bodily imperative to run because of, say, some bodily discomfort and remain near the dog. And that is a prima facie reason to run. But this gets the explanation wrong, I think, of why one runs. Because I'm going to run away from the dog because the dog is dangerous, not because running away will relieve bodily discomfort. And I think that this is an endemic problem for their view. One cannot simply appeal to pleasure or pain or comfort or discomfort to explain why emotions push and pull us. The source of motivation needs to be evaluative, and then that evaluation needs to be about the objects themselves. So this directly motivates a perceptual theory of the emotions. And on such a theory, an emotion is just a perceptual representation of an object's evaluative properties. So in the case of my fear of the dog, that is a perceptual representation that the dog is dangerous. And this can directly explain why emotions give us prima facie reasons to act and judge in certain ways. And I think of this as perceptual largely for epistemic reasons, in the same way that sensory perception is plausibly reasons indicating because it has sensory content that reveals things about the world. Likewise, emotions are plausibly reasons indicating because of their evaluative content. To summarize, the issue with the neo-Jamesian theories of emotion is that they can't explain how emotions motivate and rationalize judgment and action. I think a promising alternative is a perceptual theory of emotion, where emotions are perceptual experiences that represent evaluative properties. Thanks for listening. I look forward to your questions and comments.
Hi there, my name is Peter Grindrod. I'm Professor of Mathematics at the Mathematical Institute at the University of Oxford. And today I'm going to talk about cortex-like complex systems, what occurs within. Imagine a vast system of excitable and refractory neurons, 10 billion of them, and each individual neuron itself is excitable, meaning if it's perturbed with incoming signals, it can spike itself, and it's also refractory, meaning that if it's spiked just now, it can't spike again for a little while until all the neural chemistry becomes back in balance. The network itself is not a random network. We know quite a lot about it. It's highly modular, and the modules correspond to tightly connected parts of the neural network, 10,000 or so neurons within each module, heavily connected. 50 or 70% of the connections are present in the neuron-to-neuron connections within columns, within modules. But then the modules themselves are arranged in a kind of two-dimensional sheet where neighbouring modules are kind of connected up loosely, and that sheet itself is corrugated up over the cortex, over the outer brain structure. So what do we have? We have the excitable refractory neurons themselves, we have the architecture which is highly modular, and we have some time delays. So if I'm in one module and I'm connected to a neuron in my own module or a different module, it's going to take some time for my spike to reach the recipient spike, and then they'll spike or not, depending on whether they're in a refractory period or whether they're excited enough. So that means that the whole system is really like a massive delay differential equation. If you throw in some stochastic elements, it doesn't make anything more difficult or improve matters. And the central question is when you have such an architecture and when you have such active and reactive nodes within such a network architecture, then what type of things usually occur within? Is it just a deterministic system that takes input stimuli to output actions and things like that? Or does it possess an inner life? I'm going to argue for the latter. Now, we can't reverse engineer a human brain because that would require getting down to monitoring 10 billion individual neurons. But we can reverse engineer a synthetic brain. So what we've set out to do at Oxford is to create a synthetic network climbing up to 1 billion and then 10 billion neurons, and put them within the architecture that we know to be present in cortex-like complex systems, and have parameters controlling the excitability, the refractive nature of neurons, as well as the time delays, the times it takes for signals from one neuron to reach another. And we can simulate all that. We don't have to build a computer to do that. Those computers already exist. And in particular, we can use computers with 1 million cores to enable us to simulate these vast delay differential systems. Now, what do we see when we do this? Well, we created such a system and we stimulated it in many, many different ways. That is to say, we picked a particular input neuron and just kept pinging it with some kind of signal going in, and then we look at how the whole responds. And then we pick a different neuron and we ping it there. We see how the whole system responds. And we carried out 1 million such experiments. What would we see? Would all the responses be the same? Would all of them be just passive responses, or would they be much more active and have a structure of their own? Well, the latter is true. The way we did this was to carry out such experiments and to watch what happened at a large number of neurons, synthetic neurons, when particularly there was an input neuron that was stimulated, and then do the experiment over and over. What did we find? Well, we could characterize the functional response in the case of each experiment, and we did that as follows. We looked at the firing across the brain, and we looked at the phase differences in the firing, so that we were able to take absolute time out of it and just talk about phase patterns. So these are dynamical patterns over time, absolute time doesn't matter, of repeated firings going across the synthetic cortex-like complex system while it's being stimulated at some neuron or another. We do that many times, and we can compare the outputs, that is, these phase patterns. To do that, we need a metric on phase patterns. And so we can come up with a kind of dissimilarity or similarity matrix. So we take all the thousands and thousands and thousands of experiments we've carried out, and we produce a big matrix showing similarities of the phase patterned response to these different types of perturbation. Finally then, we can block diagonalize that matrix, which is equivalent to clustering. What we do is we say, well, similar responses can be clustered together and they're of a class. And what do we find? We find that the complex-like system has a kind of hierarchy of internal latent class-like behaviors. So if we do 1 million experiments, we don't get 1 million different, we might get 10,000 different types of behavior, or more likely 1,000 different types of behavior. And each one of those types contains replicable and very similar responses to inputs. So the inner life of such a dynamical complex system really corresponds to a whole load of latent classes. They can't be seen from outside, they can only be observed with inside. And each one of those classes yields a certain type of dynamical behavior over time and across the brain. So what does the inner life of a cortex-like complex system look like? Well, it divides into a number of latent classes, some of them very prevalent, some of them not so prevalent, and that whole is in a kind of hierarchy. So we get highly localized, specialized classes, but some of those can be grouped up to be more general classes. Where do feelings come from? What is it like to be a human brain? Well, the experience of being a human brain must be to possess these internal classes, and then sensations of one class or another being locked in, being dominant at a time, are what it is like to be a human brain. Thus, if we have the architecture, if we have the chemistry, if we have the physiology of a human brain, so necessarily you're going to have these kind of latent classes of behavior, and the brain will enjoy a whole range of sensations appearing in one-to-one correspondence with those classes. So these classes are good candidates for qualia and feelings, aren't they? And sadly, we're going to have to deny the existence of philosophical zombies, because zombies are tricky, aren't they? They have all the stuff that we have, but they don't have feelings. But actually what we're showing here is that if you have all the stuff that we have, if you have the architecture, if you have the neurons, if you have the dynamics, if you have the time lags, then necessarily you have these inner latent classes which we're kind of arguing are in one-to-one correspondence with feelings. Now, the feelings would give a fast-thinking evolutionary advantage to brains, because once you've got one of these modes, one of these internal latent classes locked in, it kind of reduces the problem set for any immediate decision making. and reduces the domain of possible perceptions, what's going to happen next. Instead, what you have is a way of limiting the decision set, and therefore there's a sort of fast thinking advantage, pretty much exactly in the way that Kahneman describes in his Thinking Fast, Thinking Slow thesis about how an efficient brain can use shortcuts, can use heuristics in order to be agile and to make rapid decision makings. So at a local scale, the internal, I'm going to call them modes. It's not the same as brain modes as functional modes, as was studied in Berlin more than 10 years ago. These are internal modes, these are latent classes of behavior. And each of those is dominated within a few modules and corresponds, those are columns in the cortex, and correspond to internal states and sensations. And I'm arguing that these are what philosophers call qualia, and the system's own experience as being in such an information processing mode. There's an evolutionary advantage to developing them, and so our brains have developed them. And as a kind of side effect of that evolutionary development, we've recognized these internal modes as sensations, and we experience the brain being in those modes. Over a large scale, then some of these internal sensations and feelings might become much bigger, like emotions, greatly limiting immediate information processing, what's going to happen next. Now, what we've shown in our work is that such internal latent modes certainly exist in Cortex-like complex systems within very large-scale simulations. That creates its own big data problem, actually, because when we reverse engineer such large systems with 1 billion or up to 10 billion nodes, we have invented a big data problem, where, remember, in each experiment we want to classify the kind of phased firing behavior going on across the whole brain, and then we want to compare those experiment-wise to say, well, how many classes have we got, how different are they, and things like that. But this is all doable. This is work for data science. And the fact is that we can now reverse engineer such large-scale simulations, and we can see what occurs within. There are latent dynamical modes, and having those, experiencing those, is what it is like to be a human brain. There is nothing more. Thank you.